Biology 1010, presentation number 7, cellular basis of inheritance. This presentation is going to largely concern organisms that have a sexual life cycle. And to begin with, we need to understand the difference between what is diploid and what is haploid. Some cells have two sets of chromosomes. For example, you and I, our individual body cells, have chromosomes arranged in pairs. Those pairs are called homologous pairs. You will see to the illustration at right a cell designated 2N, which indicates that cell has two sets of chromosomes. The chromosomes that are illustrated there are equivalent. There's two of them. You have inherited one from your mom and one from your dad. Hence, at the top of the illustration, you'll see an egg cell and a sperm cell, and the result is the brand new diploid cell called a zygote, which ultimately would divide to form you or me. Now, some cells have only one set of chromosomes, and those would be those sex cells that are illustrated above. The chromosomes occur singly, and the cell is designated as 1N. In the case of 1N or 2N designations, the N refers to copies. So I have one copy of each chromosome in each cell, or two copies of each chromosome in each cell. And you can see that in the illustrations at the very bottom right of this particular slide. As stated while we were looking at the previous slide, diploid cells have chromosomes arranged in pairs. And in animals, that would be the zygote or the fertilized egg and the cells of the body that arise from cell divisions of that egg. Egg is fertilized, it divides, and it becomes a multicellular organism. Every single one of those cells will have two sets of chromosomes. The same would be true for plants. The seed contains an embryo that will grow into a plant consisting of diploid vegetative cells. On the other hand, haploid cells containing one chromosome each will be found in the sperm of animals, the eggs of animals, and the fertilization of the two results in the zygote. Similarly, when you look at plants, they have pollen, which is the male part of the plant life cycle, and ovules, which is the female part, and the combination of the two through an act of fertilization gets you a diploid seed that grows into a brand new diploid plant. Sexual life cycles are characteristic of all eukaryotic organisms. In other words, if you're an animal, if you're a plant, if you're a protist or a fungus, you are going to have a sexual life cycle. Bacteria or prokaryotes, on the other hand, do not. So diploid and haploid cells are a characteristic feature of a sexual life cycle. Fertilization is a process by which two haploid cells, commonly called sex cells, fuse and form a zygote. If you take a look at the human life cycle illustrated at right, you will notice that humans produce sex cells, also known as gametes. And those gametes are either sperm, haploid, or eggs, haploid. And the two come together through a process we commonly call fertilization, but I certainly would like you to understand that the official term for fertilization is the word syngamy. Prefix syn means together, gamy referring to gametes. So fertilization or syngamy is a process by which gametes come together. Now there's one other thing I want you to note about this human life cycle illustrated at right. You will notice that fertilization is identified in a yellow box on the right-hand side of the cycle, and it signifies a transition from what is haploid to what is diploid. Now, on the left-hand side of the cycle, you will see a similar yellow box labeled here meiosis, which also represents a transition, in this case, from diploid to haploid. Meiosis and fertilization are two critical components of a sexual life cycle. You cannot have a sexual life cycle without fertilization or syngamy, and you cannot have a sexual life cycle without meiosis. Well, let's focus on this thing called meiosis. Meiosis is sometimes also referred to as sexual cell division. So, by definition, meiosis is a process by which sex cells or gametes are made. You take a look at the illustration at right, you will see that we start with a diploid parent cell. And that diploid parent cell appears to go through two separate divisions in sequence. You'll see the production of two products, and then from those two products, you'll see a production of a grand total of four products, each of which are haploid. Meiosis begins with a diploid parent cell and produces, as a rule, four haploid daughter cells. Those haploid daughter cells might be sperm cells, and those haploid daughter cells might be egg cells. More specifics about human sex cells in just a little bit, but let's take a look at meiosis in terms of how it fits in the general sexual life cycle. You'll remember that I mentioned on the previous slide that meiosis and syngamy seemed to be points in the life cycle where you alternated between what was haploid and was diploid, or between what was diploid and what was haploid. Well, in the case of meiosis, meiosis' job is to counter the effects of syngamy. If you think about it, syngamy, being a process of fertilization, is going to tend to multiply or amplify chromosome number. And that would be ridiculous year after year for an annual plant, or generation after generation after generation for humans. You might go from diploid individuals to tetraploid individuals to octoploid individuals to individuals with 16 sets of chromosomes, etc., etc. Every single generation, what we really need to do is reset chromosome numbers so we never get beyond 2 sets of chromosomes. So the purpose of meiosis is to counter the effects of syngamy, and its job is to actually reset chromosome number after every event of syngamy. Meiosis produces sex cells or gametes which are haploid, and when they fuse, then you get a brand new diploid organism, and then that diploid organism produces sex cells through meiosis, and the process of syngamy results once again in a diploid organism, and we never get beyond 2 sets of chromosomes in any one generation, and that's a good thing. Let's take a bird's eye view of meiosis. This particular slide shows me, once again, that meiosis is actually a 2-step process of division. The first step is called meiosis 1, and the second step is called meiosis 2. Two very distinct things happen during these steps. Now, you'll remember the chromosomes are arranged in pairs. So the very first thing we got to take care of is to separate the pairs. That's where it says meiosis 1 involves the separation of homologs. We'll see this in detail on the next couple of slides. Once we separate the homologs, then we got to take care of separating the chromatids. The chromosomes consist of 2 sister chromatids, and we're going to have to pull them apart. And that's where meiosis 2 comes in. Meiosis 1, separation of homologs. Meiosis 2, separation of chromatids. The following series of slides will look at the process of meiosis in detail. Now, you could probably do a Google search of meiosis and wind up clicking on images and getting hundreds of illustrations of various stages of meiosis. So the illustrations you'll see on the following slides are nothing more than my choice of illustrations. You could go ahead and review illustrations online to your heart's content. And maybe some of them will help you remember, and maybe some of them will not. Let's take a look at the process. This slide shows me a diploid cell that is in interphase. Now, you'll remember interphase is a term that describes the appearance of a cell that is betraying no activity whatsoever. So I see a cell, I see a nucleus, I see what is probably DNA or chromatin inside of that nucleus. It's not condensed. I don't see chromosomes. I see nothing going on. Now, in human beings, that might be a diploid cell in a man's testes called a spermatogonium, which might ultimately produce sperm. Or in a female, it might be an oogonium, a diploid cell that could ultimately produce eggs, and that would be found in the ovaries. What is convenient about the process of meiosis is we're going to use the same terminology to designate the different stages, much as the same as we used for mitosis. You'll remember that in mitosis we've got prophase, metaphase, anaphase, and telophase. We will use those same terms, but in this case we will designate them as either prophase 1 as occurring during meiosis 1 versus prophase 2 as occurring during meiosis 2. So let's take a look at the process from the start. We had just mentioned we begin with a diploid cell in interphase. So we first move into something called prophase 1. Now, prophase 1 will include typical prophase events, the same events that we associated with mitosis. I see here the chromosomes condense. The nuclear envelope is going to deconstruct because we don't need it getting in the way since we're going to be moving these chromosomes apart. And then we get this thing called a spindle. And you remember the spindle is a microtubular structure whose job it is to pull those chromosomes apart. Now, the one thing I want to notice on this slide is you'll see that we have our chromosomes are actually arranged in pairs. I see 2 big ones, I see 2 small ones. In addition, if I look more carefully at the picture, I will see what looks like the blue and the red chromosomes seem to be tangled up a little bit. And that's a very, very important part of this prophase 1 step of meiosis. The process is called crossing over. It is unique to meiosis. It occurs only in prophase 1. And on the next couple of slides, we're going to take a look at exactly what this process of crossing over is all about. Well, let's take a look at crossing over. On the left, I see a blue chromosome, maybe inherited from my dad. I see a red chromosome, maybe inherited from my mom. And I notice that those chromosomes occur together, which would make sense. They are a homologous pair. They represent the exact same chromosome, and even though that the genes in the chromosome on the left and the genes on the chromosome on the right might be a little bit different, they are exactly the same. So for example, my dad may have contributed a gene for brown eye color. On the other hand, my mom might have contributed a gene for blue eye color. And those genes are specifically different, but I still have an eye color gene on each of those chromosomes. Regardless, in a diploid cell about to undergo meiosis, during prophase 1, the chromosomes cross over. And you'll see a piece of the blue chromosome line over a piece of the red chromosome. 2 very important operative words you might have scribbled down next to the slide on your notes. And that's the word break, B-R-E-A-K, and the word reattach. So you'll notice that on the very far right hand side of the illustration, my blue chromosome has a little piece of red on it, and my red chromosome has a little piece of blue on it. And that means I've actually exchanged genes between those 2 chromosomes. Think of it as almost like shuffling cards in a deck. You have all the cards arranged in their suits. The red suits, the black suits, and they might be arranged in order, but the fact of the matter is when you shuffle that deck of cards, there's no guarantee where any one card's going to wind up. And in this case, what we're doing is we're actually shuffling genes between these 2 chromosomes. That's why I've got the word genetic exchange down below. The basic point is that when sexual reproduction occurs, you get an offspring. And that offspring, while it might resemble mom and it might resemble dad, in some cases might not perfectly resemble either one because the genes get shuffled in that process of producing the egg or the sperm that ultimately leads to form that particular offspring. So we've been through prophase 1. The chromosomes condensed as pairs. The nuclear envelope degenerated, the spindle formed, and then we had this process of crossing over taking place. Now, at this point, we move from prophase 1 into metaphase 1. And I see our chromosomes are still paired. They line up. Much like the process of metaphase in mitosis, they line up at that imaginary plane in the middle of the cell that's called the equatorial plate. And looking at the picture here, you'll notice that the spindle is there all ready to start moving these homologs apart. Because you'll remember, meiosis 1 is the separation of homologs. And so of those paired chromosomes, the red one will move in one direction and the blue will move in the other. Anaphase 1, once our homologous chromosomes line up, they pull apart. Should be no surprise. Anaphase in mitosis resulted in the chromosomes separating and moving to opposite poles of the cell. I see the same thing happening here. One member of each homologous pair is pulled by the spindle to opposite poles of the cell. At this point in the process of meiosis, the cell is going to take a little bit of a break. Now, you'll remember in the process of mitosis, telophase was basically prophase in reverse, where we decondensed the chromosomes, we deconstructed the spindle, and we reconstructed a nuclear envelope. Well, at this point, it's a little bit different. As a matter of fact, telophase is significantly different at this point than it was in mitosis. Telophase 1 is almost a non-telophase because we still have another division to complete. We've done with meiosis 1, but now we need to go into meiosis 2. So there's really no point in decondensing the chromosomes. So I see here the chromosomes stay condensed. I don't need nuclear envelopes to get in the way, so the nuclear envelope does not reform. Now, the spindle will deconstruct, but the only reason the spindle deconstructs is I need to put it back together again to accomplish the next division process. And I'll have to reconstruct it in a different orientation than previously. So this is kind of like an intermission, a break in between meiosis 1 and meiosis 2, and it's sometimes also referred to as interkinesis. It's a break, a slight pause between meiosis 1 and meiosis 2. And the last bullet point here acknowledges that sometimes we might have some cytokinesis going on. I see the cells essentially splitting there, but not completely. But sometimes that might not happen at all. What we're about to do is take a break and then go straight into meiosis 2. Okay, so meiosis 1 went through prophase 1, metaphase 1, anaphase 1, telophase 1, and then interkinesis. Let's go through meiosis 2. We're going to go through prophase 2, metaphase 2, anaphase 2, and telophase 2. Our picture here shows us the first step of meiosis 2. It's called prophase 2. Typical prophase events, but remember my chromosomes are still condensed from before. There still is no nuclear envelope. Our spindle reforms so we can actually move these chromosomes in a different direction than we did before. Got to remember though that there is no crossing over during prophase 2. But remember that crossing over was exclusively part of prophase 1. It is not part of prophase 2. Metaphase 2, you'll be gratified to realize that this is not terribly different from what we would expect. The chromosomes line up at the equatorial plate in anticipation of anaphase 2. Anaphase 2, once my chromosomes have lined up, the individual chromatids are going to separate, and those chromatids move to the opposite poles of the cell. Telophase 2, we are now finished with the process of meiosis. So telophase 2, we no longer need our chromosomes, so they will decondense. We no longer need the spindle. It will deconstruct. And now I've got 1, 2, 3, 4, count them, products. And every single one of those new products is a brand spanking new cell. And so the nuclear envelopes need to reform at this point. Telophase 2 is essentially prophase in reverse. It's accompanied by cytokinesis, and the result is going to be 4 haploid products, each one of those products containing a single copy of one of the previous chromosomes. Right, so now we have a real good general idea of how meiosis proceeds. Let's take a look at how it proceeds in detail in humans. Understand that meiosis is a process of sexual cell division that results in the formation of sperm in males and eggs in females. In males, sperm formation is commonly called spermatogenesis. The word genesis meaning creation. So it's creation of sperm. It occurs in the testes, which are gonads. It occurs on a daily basis. Inside of the testes are some very, very tiny tubes called seminiferous tubules, and those seminiferous tubules contain the diploid cells that will produce the haploid sperm cells. Now, in males, it is a typical process of meiosis. In other words, the process will resemble the process that we just looked at. It will result in 4 viable or functional products for sperm cells. In females, the process of egg formation is called oogenesis. Genesis for creation, oo implying ovum, meaning egg. So creation of eggs occurs in ovaries, which are gonads, but in this case on a monthly basis. And inside the ovaries are small spherical jacketed structures called follicles, and each follicle contains a diploid cell that will undergo meiosis to eventually produce an egg. And most of you all realize that on a monthly basis, once a girl reaches puberty, those eggs are ovulated on a monthly basis. And that process is the end of the meiosis process. In the females, it's how she produces eggs. In the female, though, the process of oogenesis is an atypical process of meiosis. In other words, it's not going to resemble perfectly the general process that we just looked at. You take a look at this. In the process of oogenesis, we're going to make 3 products, not 4. And of those 3 products, only one is a viable or functional cell, and that is the egg cell, sometimes also called an ovum. Now, the other 2 products are non-viable products called polar bodies. Now, we'll take a look at the details of this in a minute. But the idea is that in the female, what the goal is to produce a single viable egg cell and make sure we maximize the cytoplasmic resources inside of that egg cell. But I still have to go through the process of meiosis. So rather than producing 4 equally sized cells with maybe 25% of resources in each one, what I'm going to try to do is generate one cell with 98% of the resources and then produce like 2 other products that aren't going to contain any resources at all, because I'm basically putting all my resources into the egg cell. Right, let's take a look at the process of spermatogenesis as it occurs in males. The illustration found on this particular slide is one of thousands that you could access on Google, just the one I chose to look at. Now, what I'm going to ask you to do is I'm going to ask you to make certain that you circle some cell types as we go through this process. There looks like there's a lot of words printed on this particular slide. There are some cells that are going to be really, really important that I need you to know. So I will tell you those as we go through this slide. Let's take a look at the process of human spermatogenesis. The first cell I want you to circle is the word spermatogonium up there at the top. Now that's a diploid cell found in one of those seminiferous tubules in the male's testes. It's a diploid cell, and a spermatogonium, it's real important to realize that it might or might not undergo meiosis. The implication is it could become a sperm cell at some point, but if you look at the illustration, the spermatogonium, while it could result in the formation of sperm cells, it could also divide and make more spermatogonia as well. And when you consider the fact that men are making sperm 24 hours a day, 7 days a week, their entire reproductive lives, spermatogonia are going to need to be pretty darn active to produce more and more spermatogonia so that this guy could be producing these sperm cells anywhere from the time that he's 12 years old maybe until he's like 82. Now the next cell down is called the primary spermatocyte, and I want you to circle that. The primary spermatocyte is a diploid cell, and it's actually nothing more than a spermatogonium that will undergo meiosis. Primary spermatocyte is diploid, but it is destined to undergo meiosis, and so I see the primary spermatocyte will undergo meiosis 1, and that will be followed by meiosis 2. The next cell type you want to circle there are secondary spermatocytes, and you'll see that is a result of meiosis 1. The primary spermatocyte undergoes meiosis 1, produces 2 secondary spermatocytes, and those then undergo meiosis 2, resulting in spermatids. Circle spermatids, those are nothing more than immature sperm cells. Now see, at this point we've transitioned from a diploid spermatogonium to a diploid primary spermatocyte to haploid secondary spermatocytes to haploid spermatids, and those haploid spermatids will mature into sperm cells. Now you'll notice they use the archaic or old time term spermatozoa. Hardly anyone who's alive anymore still uses that term, so we're just going to call these sperm cells. So in summary, I'm going to look at spermatogenesis, and I'm realizing it is a typical process of meiosis. I start with a cell called a spermatogonium. It might or might not undergo meiosis. If it does, it becomes a primary spermatocyte, undergoes meiosis 1, produces 2 secondary spermatocytes, undergoes meiosis 2, produces 4 spermatids, which at this point are haploid, and those 4 spermatids mature into 4 haploid sperm cells. Now, I know that previous slide took a long time to get through. On this particular slide, all I did was I made a list of those same cell types that I asked you to circle in the process of spermatogenesis. This will be a great help in combination with the previous illustration. Well, let's take a look at oogenesis. You'll remember I said that it was an atypical process of meiosis. What we're going to notice is that the same cells are listed here. They're just a little bit termed differently. It's because it's a process of oogenesis. So instead of spermatogonium, I see an oogonium. Instead of a primary spermatocyte, I see a primary oocyte. But you'll also notice that the process, as illustrated, looks a little bit different from what we've seen before. Now, I will make a very important note here. If you Google oogenesis, you need to be very, very careful because there are some, let's say, inaccurate illustrations of oogenesis out there on the web. You will want to be sure that you will be looking at an illustration of oogenesis that results in the formation of one egg cell and 2 polar bodies. In many cases, there will be some illustrations of oogenesis that have 1 egg cell and 3 polar bodies. That's not what you want. So let's go ahead and run through the process. We'll try to be a little more quick this time, and I will just foreshadow the fact that the next slide in the presentation will have these same cell types labeled. We start off with an oogonium, which is a diploid cell located in a follicle in an ovary, and that oogonium might or might not undergo meiosis. It might or might not result in the formation of egg cells. If the oogonium is going to go through the process of meiosis, it becomes transformed into a primary oocyte, and the primary oocyte will then undergo meiosis 1, which will then be followed by meiosis 2, which will result in the formation of an egg cell. The primary oocyte is diploid. It undergoes meiosis 1. Now look very carefully. The process of meiosis 1 results in 2 products that are unequal in size. The one on the left is called the secondary oocyte. That's viable. It will continue on through meiosis 2. But right next door to that secondary oocyte, you'll see a smaller thing called the first polar body. It is nothing more than a teeny tiny little product containing 1 set of chromosomes because I need to separate the homologs, remember? And I don't need the homologs in that particular polar body because what I'm trying to do is I'm trying to maintain all of my cytoplasmic resources in that secondary oocyte. So unequal cytokinesis in meiosis 1 results in 2 unequal products, a secondary oocyte and a polar body. The secondary oocyte will continue to divide, goes through meiosis 2, forming an egg cell or an ovum and a second polar body. Once again, we're trying to maintain maximum amount of cytoplasmic resources in that egg cell. So all told, process of oogenesis begins with an oogonium diploid, which may or may not undergo meiosis. If it does, it becomes a primary oocyte, which goes through meiosis 1, resulting in 2 unequal sized products, a secondary oocyte and a first polar body. And the secondary oocyte undergoes meiosis 2, resulting in an ovum, egg cell, and a second polar body. Those 2 polar bodies are non-viable. Realistically, what happens is through this process, the polar bodies eventually die and are reabsorbed into the ovary. And so the resources contained in those polar bodies, even though there's not much of it, are not left to waste. And as a result, that ovum down there at the bottom of the screen contains virtually 98% of all the cytoplasmic resources contained in that original oogonium. And certainly from the standpoint of survival of the offspring, you want to have as much cytoplasmic resources in that egg cell as possible, because realistically, sperm cell doesn't contribute anything at all short of 23 chromosomes. Sperm cell has no cytoplasmic resources to contribute. So basically, the egg has to take all of the responsibility for the future success of that fertilized egg as it develops into an offspring. So, as promised, the following slide is nothing more than a list of all of those cells you need to be aware of from the process of oogenesis. Oogonium, primary oocyte, secondary oocyte, first polar body, ovum or egg cell, and second polar body. And this slide works very, very well with the previous slide in terms of getting a good idea of what the process is involved. Our last few slides in this presentation are going to deal with problems with meiosis. The first problem is something called aneuploidy. Now, aneuploidy can involve having extra chromosomes, as shown on this slide, or having missing chromosomes, on the next slide. In this case, an individual could, in fact, inherit 3 copies of a chromosome, and that would be referred to as a trisomy. If you take a look at the young man illustrated at the top of the slide, you will see that his karyotype, which is nothing more than a visual representation of his chromosomes, you see he's got 3 copies of chromosome number 21. That is a classic example of an aneuploidy. It's called Down syndrome. In that case, his mother probably ovulated an egg that contained 2 chromosome number 21 instead of just 1, and that was fertilized by a normal sperm cell that resulted in the formation of a zygote with 3 copies of chromosome number 21. Now you will notice below it says this is caused by something called non-disjunction. You probably might want to write down that non-disjunction is a failure of chromosomes to separate during meiosis. Another disorder that is involved in aneuploidy or having extra chromosomes is Klinefelter syndrome. You'll see the individual at right there at the bottom is a phenotypic male. In other words, it is a male individual, but that male individual has 2 copies of the X chromosome. So instead of being a normal male, which is XY, the individual's XXY. And so you see he has inherited 2 copies of the X and 1 copy of the Y. The Y was contributed by his father, and the X's would have been contributed by his mother. Once again, probably a case of non-disjunction or failure of chromosomes to separate, and that would have happened during her formation of that egg. I will tell you this, as women get older, the incidence of non-disjunction gets increasing. So if you got a woman who ovulates at the age of 18, chances are that egg is going to be perfectly fine. But if you got a woman who's ovulating at the age of 46, there's a very, very good chance that the egg may have experienced a case of non-disjunction during its formation. Still a pretty rare thing, but still worthy of some concern. As a rule, older women have greater chance of problems associated with failure of chromosomes to separate during oogenesis. You remember that I said that aneuploidy can be having an extra chromosome, or you could be missing a chromosome. Missing a chromosome results in something that is commonly referred to as a monosomy, and in this case, you see the young lady at right is a person who has something called Turner syndrome. Now, in Turner syndrome, you'll see that she is, for all intents and purposes, a phenotypic female, but if you look at her genotype, you'll notice that she has 1 copy of an X chromosome instead of 2. You would designate her not as XX, but basically as X0. But once again, in this case, it's probably a case of non-disjunction, in which case the X that she has was probably contributed by her dad's sperm, but her egg cell might not have contained any X chromosome at all due to a non-disjunction in the process of oogenesis in her mom. Aneuploidy, either extra chromosomes or missing chromosomes, and make sure you associate these different disorders with that. Down syndrome, having an extra chromosome. Klinefelter syndrome, having an extra chromosome. Turner syndrome, missing a chromosome. The details on this slide are not critical. It's just nothing more than a review of what non-disjunction is and gives you an opportunity to look at what it looks like in the process of meiosis. It's a failure of chromosomes to separate during meiosis, and I said a lot of times this happens in older females, but if you take a look at the process of meiosis below, I see that it could occur during meiosis 1, it could occur during meiosis 2. In other event, if we take a look at the gametes produced, you might have an extra chromosome or you might be missing a chromosome. And it's important to remember that that is going to have an effect on that egg cell if it winds up getting fertilized. And so the egg cell missing a chromosome is going to result in an offspring that's going to be missing a chromosome. An egg cell with an extra chromosome is going to wind up leading to an offspring with an extra chromosome after an event of fertilization. Now, while aneuploidy might seem to be a disorder, there is a problem with meiosis that is actually part of the natural scheme of things, and that's something called polyploidy. Polyploidy means having multiple sets of chromosomes. It's a problem that's fairly common in plants, and I guess you might not say it's a problem because in some plants it actually increases reproductive output. You take a look at the strawberry over there on the right. Normal wild type strawberries are generally diploid. But those big giant strawberries you buy at Walmart, as a rule, are octoploid. Those strawberries have 8 sets of chromosomes instead of 2. But the thing is, in the plant, having 8 sets of chromosomes will increase fruit size and, as a rule, doesn't have a huge effect on the overall health of the plant. On the other hand, in animals, it's a relatively rare thing because in most animals, increasing the sets of chromosomes can have negative effects on the ability of the individual to survive. So polyploidy is a natural phenomenon. The examples are not important there. Essentially, there are some plants that are polyploid naturally. In some cases, polyploidy can be induced, like the strawberries there, or you'll see there is one animal example, the grass carp below, which is a triploid individual. It's one of those rare animal species that is not negatively affected by the number of chromosomes it has, but you take a look at the karyotype that's photographed at the left, and you'll notice it's got 3 copies of each chromosome. Make sure you know what polyploidy is.